Let's see.
Sounds good.
honestly, my sentiments for who I'm going to be
online people, can you hear me? If you can hear, can you type in the chat or just speak? Wonderful, wonderful. Thanks. The microphone works. No problem.
Okay.
Yeah.
So everybody hear me okay? I'm testing my microphone here. Hello, hello, hello.
It works.
Thank you.
Yeah. Wonderful. You have your file. That's your file, right?
It's
Your 10. This one.
this is a robotic arm we purchased.
I see it's like it was a robot. 10.
Yeah.
How the breaker is do you have any factor here? Or it's just like
That's our sensor.
camera?
Yeah.
I'm going to show some demos like videos like that. One for one. Okay, we're gonna get started.
Okay. Give me 1 second. I think it's better to still duplicate. Good. Okay.
We can sort of see both sides.
Yeah.
Okay, wonderful. Let's get started. Yeah. Just see. I think people can hear us. Great. Hello, everyone. Thank you here online, and also thank you people coming here for our last event for this last ICON weekly seminar for this semester. I'm I'm assistant professor from industrial engineering and working in robotics. And today it's a pleasure to have Professor Jian here to talk about his research. And actually, for the last event, we have actually kind of 2 sessions for the seminar today. Professor Jian, he will talk about the research in the first part. And if you're interested in music, actually he brought a guitar. He will do some guitar playing after the seminar.
It's holiday season and it's Friday afternoon. Why not?
If you're interested in music, you're more than welcome to stay, enjoy the music. Okay, let's get started. I will briefly introduce Dr. Jian. So he is an associate professor from Department of Agriculture and Biological Engineering, and he received his PhD degree from Agriculture Engineering from Iowa State, and he received the master degree from Denmark Technical University, and also a bachelor degree from Zhejiang University in China. And his major research interest is to develop the next generation plant phenotyping technology, along with machine vision, image processing, and data modeling. He also interested in other areas of agriculture, sensing broadly defined in automation and robotics in agriculture. Without further ado, let's welcome Professor Jian for the seminar.
Thank you very much for the introduction. Hello to everybody in the room and everybody on Zoom. So it's great to be here, and thank you for the invitation again. And now it's holiday season, and it's a great Friday. And last week was the Thanksgiving holiday, and I hope everybody enjoying some great food with your family and with your friends, right? Yes. However, I want to remind you, right now in the world, 800 million people are still suffering from hunger right now. And I was driving to work just a couple of weeks ago. I was listening to the radio, and anybody noticed that our world population just reached 8 billion just 2 weeks ago. And we know by 2050, our world population will reach 10 billion. So the population is growing, and in order to feed so many people, we need to produce more food. Unfortunately, our land area is not increasing. So what does that mean? That means we have to adopt new technologies to improve our food production efficiency. Makes sense, right? So I think this seminar series is from ICON. So in ICON, we talk about technologies like sensors, robotics, optimization, internet, and so on. So today, today's topic is about agriculture. So I just prepared a lot of the pictures and the videos just to show you how we can apply these technologies in agriculture, so we can help people to improve the farming and the food production efficiency. And hopefully we can do something to help to keep the world in peace. So, well, next slide. A little bit about myself. Let me move this bar a little away. Okay. So this is my brief journey. So I was originally from China. So I was born and grew up in a city named Nantong in China. which is very close to Shanghai. It has 1 million people in downtown, but we still call ourselves from a small city because we are right beside Shanghai, which is having 20 million population, right? So when I was 19 years old, I went to Hangzhou city and attended Zhejiang University and got my bachelor's degree in computer science. Just one highlight of that city. Hangzhou city is really famous for this West Lake here. Many people say this is the most beautiful lake in China. So if you, in the future, go to any area around China, I highly recommend that. So after my graduation, I went abroad. First stop was in Denmark, Copenhagen. So I attended Denmark Technical University and did my master's degree in computer engineering. So 2 years later, I traveled here to the United States, did my PhD at Iowa State, stayed in Iowa for another 5 years, working in a company called DuPont Pioneer. The company's current name is Corteva. How many of you know Pioneer and Corteva? So for those of you who are not familiar, Pioneer and Monsanto, these are the two biggest seed companies in North America. So, you know, in North America, talking about agriculture, what do we produce? Probably the top two are corn and soybean, right? So talking about selling the seeds, the seed market in Northern America, Pioneer and Monsanto, these two companies shared the 70% of the total market for the corn and soybean seeds. All the other smaller companies share the remaining 30%. So Pioneer is really, really a big company, and I worked there for 5 years until 2015. I returned back to academia and joined Purdue as a faculty member here. So for today, for the remaining 1 hour, this is my plan. Since we are going to talk about phenotyping, first of all, many of you may be interested to learn what is the progress, recent progress at Purdue. So I just want to introduce Purdue's newly constructed automatic phenotyping facilities. So I will just go through them one by one, why they are built, how we're using them, and what can we do with those facilities as well. So basically talking about the phenotyping capacities with those phenotyping facilities. But then we always say, however, right? However, I also want to share with you a lot of my personal insights of the problems and the challenges. So I will share the advanced fancy technologies first, but nothing is perfect in this world. What are the remaining challenges? Why the phenotyping sensors are still not adopted by farmers? Farmers are still using their eyes or just sample the leaf, send to the wet lab for assessment of the nutrients and the diseases. Drone companies have been started up. Hundreds of drone companies have been started up. But so far until today, we haven't seen any drone imaging companies growing up to the size we expect them to be. Why is that? So I'm going to share some of the technical insights and try to explain the reason why they are still not adopted in practical farming. And of course, we don't want to leave you in a miserable feeling with all those challenges and the problems. So of course, in the final part, I'm going to introduce the Purdue solutions to those challenges. Why we believe Purdue may play a very unique role in this one to solve those issues and push the phenotyping technologies into the practical agriculture production process. So first one is the Purdue's automatic phenotyping facilities. This is like a summary, a gallery of what we built so far. So how many of you heard about Purdue's research initiative called Plant Science Initiative? This is engineering college, so I understand. So just some background. I think it's about 8 years ago, President Mitch Daniel allocated about $20 million to initialize some research activities in plant phenotyping and plant sensing. And especially over the last 5 years, it has been very, very exciting 5 years. Because over the 5 years, we deployed 4 new crop plants phenotyping facilities, including some of the greenhouse phenotyping facilities, the field phenotyping facilities, and some indoor facility with growth chambers and imaging stations. I'm going to go through them one by one, but before I do that, I realize that in this room, many of you may be lacking some background about phenotyping. So first of all, very brief introduction of the history of phenotyping. What is phenotyping? So phenotyping is basically, you can compare phenotyping with genotyping. When people do genotyping nowadays, we are still in a pandemic. Some of you are still wearing masks. So, when we want to test if the COVID-19 virus, for example, is that existing in my body or not, we do the test, right? So basically we get some tissue and we can analyze basically the DNA, the information of the DNA, and we can check the DNA, we can check the gene, and we can measure if it is this virus or that virus. That is called genotyping. However, genotyping is just the reason. Sometimes we need to know the result. So for the crop plants, we need to know, are the plants growing happily? Are they growing bigger? How many leaves are they? How green are they? Are they resistant to diseases? Are they resistant to drought stresses, nitrogen deficiency, and a lot of the pathogen diseases and insect diseases? So we want to measure how the plants look like. We want to measure this result. Basically, we want to measure how the plants show up to be. So that is called phenotyping. Basically, phenotyping is just to apply all kinds of different sensors to measure the plants and to assess how well the plants are growing. This is called phenotyping. And phenotyping really started from Netherlands. From about 20 years ago, and Netherlands is very famous for tulip flowers, right? If you have been Netherlands in the tulip flower field, beautiful thing in the spring. So about 20 years ago, the Dutch people had this idea. Can we build some automatic imaging machine like this to do the high throughput tulip flower imaging? So we can do the image analysis for each of the flowers, and we can check if my new flower variety is showing that color I wanted it to show or not. If it's not showing that color, we throw them away. So basically, this is a screening machine to screen the tulip flowers and to help them to select the better looking tulip flowers. That really started from the Netherlands. And then, of course, the people in agriculture realized the value of this kind of imaging system. Why don't we apply this type of systems also to corn and soybean plants? So something like this, you see the plants are sitting on the conveyor system. They are pushed through the bright tunnel. And in this tunnel, actually, we put cameras and the lights. It's just like a studio, automatic imaging studio. So when the plants goes by, we take these images and we can do all kinds of imaging processing. We can count how big, how tall, how many leaves, how wide is each of the leaf, things like that. So since then, people have been building a lot of the plant phenotyping facilities in North America, started from some of the major industry companies, including Monsanto and Pioneer. So Monsanto was the first company who built something like this. I think the inside picture is still confidential, so I'm just using this picture to show you the idea of the phenotyping facility. So in the facility, you grow a lot of the plants up, and each part may be containing a different gene or a different trait. And then, we use all kinds of, this machine looks like a CT scanner, right? And then we have those drone-like machines under the ceiling. And then we have some of the robotic arms holding the proximal sensors to approach each of the plants. Basically, we can apply all kinds of this kind of sensors to collect any type of information, the data for the plants. And the remaining thing is data-driven breeding. So let's say in this greenhouse, I just grew up maybe 20 or 100 different genes, different genotypes. And I want to check, okay, which genotype is showing the most drought resistance? Because I'm doing breeding for drought resistance. So what we can do is we can stop watering and we can collect data and see which genotypes are shrinking and showing the yellow color earlier. Which ones are still keeping green? Because we can do color RGB imaging, right? We can easily measure how green each plant look like. So basically, this is just one idea that we can do data-driven breeding with the phenotyping. And of course, as many of you can imagine, phenotyping technologies can be useful in the real crop growers field. Because if you are the crop grower, let's say you have a corn field, you grow corn every year. One question is, when should I fertilize my corn? Every year, each Indiana farmer dump a lot of the nitrogen fertilizers into the field. They spend a lot of the money, but they don't know, is that necessary for me to spend money to still dump more nitrogen in this field? Or should I be smarter to know where and when and how much fertilizer to put into the field? So obviously, as you can tell, if we can, for example, fly the drone over your field, and if we have a good nitrogen content measurement sensor carried by the drone, we can quickly show the map of the nitrogen distribution in your field. And then you only need to put nitrogen in those field area where we are lack of nitrogen. For example, that can help farmers to save the money and improve the yield and earn more money finally by the end of the year. And also for policy making as well, as you can imagine, if we do the regional phenotyping, let's say the satellite and the manned aircraft can be flying every day to cover the whole Indiana state, for example, the state government may use this information to have assessments of something like how the diseases are spreading across our field, both in forestry and in crop fields, right? So just hopefully you get the idea of why phenotyping is important in agriculture. It's really important to have the automatic and sensitive sensors for us to quickly measure how the crops are growing. So in this slide, I'm showing you just a bunch of different sensors. I don't think I need to explain through all of, each of them, because this is very straightforward. The sensors can be all the way up to the space. We can use satellites to scan the field. We can use manned and unmanned aircrafts. We have the robots moving in the field. And also we can have these proximal sensors to approach the leaf and just measure the details of the plants. Some people also even bury some of the sensors into the soil underground to measure the roots, things like those. And as you can tell, cameras, imaging sensors are probably the most preferred sensors, just because it's so easy to use and we have existing imaging technologies existing there. And it can provide a very quick, non-destructive, and very effective phenotyping. So that is why all kinds of cameras have been used in phenotyping, including 3D stereo imaging, RGB imaging, multispectral, hyperspectral, fluorescence, thermal imaging. You know, thermal imaging can be used to measure temperature of the leaf, which is correlated with the water content in the leaf, right? But however, today I want to highlight one technology, which is also my research focus, which is hyperspectral imaging, which I'm confident to say this is the #1 most popular sensing for plant phenotyping. Why is hyperspectral popular and what is hyperspectral imaging? Just want to quickly show you this idea. So you see 3 apples here, right? First one is a grayscale image. Second one is the RGB image. The third one is the heat map from a hyperspectral image. So basically comparing the first 2 images, you are familiar with grayscale, and let me use my pointer in the screen so the people on Zoom can know what I'm talking about. So let's compare the first 2 apples, grayscale versus RGB. You can see the second image is giving us a lot more information about the apple, right? Because in the first image, we can only tell between the brighter region and the darker region. That's it. But with RGB, we can tell the green leaf and we can tell the red apple. And looking at the green leaf, I know the chlorophyll pigments are there. And when I'm looking at the red apple skins, I know, oh, red means sweet. I got that information, means sugar. Why is that? Because red, what you are looking at is basically anthocyanin pigment. And anthocyanin is a sign of the maturity. And when you see red, that means the apple is matured, and matured means it's sweet and juicy. So that's a lot more information we can get from the second RGB image. But what's essentially the difference between the grayscale image and the RGB image? Basically, it's just the number of bands, right? With grayscale, each pixel has only 1 value. that is telling you the intensity of each pixel. But with RGB image, each pixel has 3 values: how green, how blue, how red it looks like. That is why the second RGB image is having a great, much more information, much richer information compared with one band. Basically, hyperspectral imaging is having hundreds of bands. Imagine that. So imagine if we go from 1 band to 3 bands and then jump to hundreds of bands. Can you imagine how much more we can see? So our human eyes are actually RGB cameras. We can only see 3 colors. Okay, so our human eyes are just RGB, but we have hyperspectral cameras, which is having hundreds of different colors for each pixel. That means hyperspectral imaging is way beyond human vision capabilities. And that is why it can see things human eyes cannot see or RGB images cannot see. So that is why, sorry, I need to move this away so you can see what I'm putting here, text. Well, I put it down here. So that is why the hyperspectral imaging has been well established to measure a lot of things. We can measure leaf moisture, chlorophyll content, nitrogen content, not only nitrogen, but all kinds of the nutrients, diseases, again, insects, pathogen, all kinds of diseases and chemical stresses. When you apply herbicides and other chemical products, we can observe the differences as well. So that's hyperspectral imaging. So that is also why we install the hyperspectral imaging systems in each of the 4 plant phenotyping facilities here. So this is the first greenhouse. In this greenhouse, we basically build conveyor systems, and also this black tower is a hyperspectral imaging tower. It has 2 hyperspectral cameras in it, 1 top view, 1 side view. And every time as this video is showing, every time the door automatically opens, 1 plant is pushed inside. And on each of the plant, we have the RFID to let the system know who is inside, right? And then we can check what's the genotype, when it was planted, how we treated this plant. And every time when the plant is finished, the imaging is finished, we can immediately do the real-time image processing, and we can measure nitrogen content, water content, diseases, and how big the plants look like. So this is the first facility. Of course, this is advanced, fully automated, advanced phenotyping greenhouse. But another highlight I want to highlight for this facility is it can actually also remove something called microclimate, which is a big issue for greenhouse plant researchers. Because as you can see from this picture here, in the greenhouse, we have the lights. And for example, this plant is sitting right underneath the light. If you have ever been to a greenhouse, if you sit right underneath the light, you feel your head top is being baked. And this plant is actually under some heat stress. But compared with this plant, it is cool. So that is unwanted variance in the greenhouse. Similarly, we have the air conditioner on the north side, we have the sunshine from the south side, and then we have the fans on the walls. These different equipments are all applying the gradient of the temperature and lighting, and they are generating unwanted differences between the plants because we are studying diseases and nutrients. We don't want this to appear in my data. So that is why we can also use the conveyor system in this greenhouse with an optimized control algorithm to move the plants around smartly. So with the minimum movement, we can kind of shuffle the plants to kind of average out the different microclimate effects. So that is just another highlight of this one. The second facility at Purdue is a very famous one. This is not part of my lab. The other 3 are all part of my lab, but this one, CEPF, now it's actually called AAPF, Ag Alumni Plant Phenotyping Facility. So it's a $6.5 million project invested by the College of Agriculture. It was deployed in 2018. So a few highlights. Basically, it's a new building, and this building is located just south of Lily Hall, south of the State Street. So actually 3 of the facilities here are all together south of Lily Hall. So we had the new building. That's why it's an expensive project. But some technical highlights. Yeah, in this room, we have 2 huge growth chambers like this. Each growth chamber is like the bedroom for the plants. So every night the plants sleep and live in the bedroom. Each bedroom can accommodate about 300 plants. But every day in the morning, we can send these plants out to have a ride, like in Disneyland, right? So we can give them a ride through the conveyor systems. So where do they go to? They are riding on the conveyor system to go to each imaging stations one by one. And in this facility, we are very proud to receive the funding to purchase the hyperspectral RGB and also the X-ray CT root scanner. So basically, when we advertise this facility outside Purdue, we always say this is the very unique facility, which we can use RGB and hyperspectral imaging to measure the canopies of the plants above ground. But we also have the CT system to scan the root underneath the soil. And the root imaging is actually very beautiful. So if you have a chance, I encourage you to go to the south part of Lily Hall, and I think this AAPF is hosting a lot of the tours every month. That's the second one. The third facility is another greenhouse, also south of Lily Hall. You know, south of Lily Hall, we have a big greenhouse complex. So this is another room. So in this room, we basically built this gantry imaging system. So the yellow gantry you can see here, this is a self-designed system. We are basically using this yellow gantry to play the role of a mock drone system. Why do we need to build a mock drone system in the greenhouse? Because we like to do experiments with drone imagers. But every year in Indiana, you know, we only have about 2 months in the field when we can really go to the field and have the crops in the field. Right now it's too cold and there is no crops at all, but we still want to do research. How do we do that? We can simulate the field in the greenhouse. So we can do something like this. We can grow the plants all through the 4 seasons in the greenhouse on the ground like this. And this gantry system is just like a drone, right? And then we can let the drone to carry all kinds of the cameras, including the hyperspectral cameras, RGB, you know, thermal, 3D. And so we can do all seasons. Throughout the season, we can do phenotyping in the greenhouse. And this is just one example of the chlorophyll distribution heat map we get from the camera, hyperspectral cameras of this greenhouse. And another highlight of this greenhouse is that it can not only do remote sensing. Sorry. It can not only do remote sensing, but can also do proximal sensing. So we can also install some robotic arms like this. So it is holding one proximal hyperspectral camera. And you can see every time it automatically uses the machine vision to detect the top matured leaf of this soybean. And we can just automatically move to the next position, detects the leaf again, and the robotic arm will hold the hyperspectral camera to grasp the leaf, close the chamber, do a linear scanning, because most of the hyperspectral cameras are push broom linear line scanning cameras, things like this. Okay, kind of cool, right? The fourth one, next one. The last one is not in campus, but in the field. So, you know, our Purdue research farm is about 5 miles northwest of the campus. If you go to Mayors and Menards, just keep driving up for about 3 minutes to the west. That's our Purdue research farm, 1,600 acres. It's a very big farm. We do all kinds of research over there. And this is an imaging gantry, hyperspectral imaging gantry in the field we built in 2019. Same idea, the gantry is playing the role of the drone system. So this drone is carrying a hyperspectral camera and keeping scanning this field underneath it. And one highlight is this whole field. So you know, basically the hyperspectral camera is this white camera kit, can travel along this trail back and forth, and for every round it takes only 6 minutes. That means every day, if you start scanning the field from sunrise, then you keep scanning until sunset, you can collect about 1,000 images of the same field. And I know many of you will ask me this question: Why do you need to scan 1,000 images of the same field again, repetitively over just one day? Just do one time. It's the same thing, right? So let me show you the reason. So this video is basically showing how it is working here, right? And I want to show you that this chart at the bottom of this one. So basically the x-axis from 31 to about 60, the x-axis is actually the number of days after planting. So basically 1 month after planting, the plant is about 1 meter height. We start the scanning every day, again, from sunrise to sunset. So every day we collect 1,000 or hundreds of images. So, and then we do this every day until 2 months age of the plants. So for the duration of 30 days, from 30th day to the 60th day, every day we collect about 1,000 images. And we check the result. So for example, in here we can check one phenotyping result, so-called NDVI. How many of you know NDVI? If you are not, NDVI basically means normalized difference vegetative index. And simply just take it home. The take-home message is NDVI is just greenness, how green the plants look like, how much chlorophyll the plants are holding. That's of course very important, right? So let's see, how is the NDVI measurement over the 1 month? You can see it's not uniform. It's keeping changing. It's not a flat curve. It's an undulating curve. That means at different time points, our greenness measurement is very different. Especially, and of course, you know, you can say this is over 1 month. Of course, the plants are getting greener and greener, right? But I want to show you this. This is just within 1 day. Just within 1 day. And if you amplify, if you zoom in into this, you can see there is a V shape, the V shape of the NDVI change over just 1 day. What does that mean? Yes.
The lighting condition affect the result. We normally have a huge sunshine affect the result.
Yeah, thank you. I can talk a little more about. So Dr. Yu's question is, basically why? Why is there a difference? So I actually, so he is asking, is this V-shape variance caused by the sunlight, the daylight changing? And as an engineer, as an imaging scientist, at the beginning, when I saw that V-shape for the first time, I had the same response as you. I said, okay, this is definitely just the sunlight changing, because drone remote sensing is using the sun as a lighting source, right? And the sun is not a stable lighting source. It is keeping changing the angle, it's keeping changing the intensity, it's keeping changing its color. You know, we're enjoying the red sunrise and the sunset, but during the noontime, we don't want to look at that white sphere in the sky. So, and actually this bottom of the V shape, by accident, it's just every day, it's just the highest solar noon time, it's highest sun angle time. So I said, of course, this is sun angle. But then, you know, you remember we still have the greenhouse imaging chamber. So let's check the greenhouse one. So we can use the greenhouse imaging chamber to repetitively scan the same plant again and again. We got the same change. There is no changing sun in that imaging chamber. It's an enclosed chamber with the ambient light blocked. It's just artificial lights still there. So we know, it should be plant, not the light. And then this is a long story. I don't think I have time to talk too much about this one, but I can share with this group that finally our conclusion is we are certain that the leaf color is really changing significantly every day. How many of you know that? Probably you don't know. I think you don't know this because our human eyes, we don't have a good memory of how the plants look like several hours ago, right? But if there is a time machine, you put just the morning leaf and the afternoon leaf side by side together, you can probably see some obviously change of the leaf color. And we are working.
So I had a question. So in the greenhouse, the lights are on all the time, so the plants don't know day and night, correct?
So when I'm talking about the greenhouse one, I'm actually talking about this facility. So in this black box, when the plant is imaged, the door is closed. There is no ambient light entering this. We are using the controlled artificial light, which is completely stable. But
no,
still,
I understand.
Yeah,
but the rest of the greenhouse, the environment is the same all day. There's no night and there's no day, correct?
So actually, the greenhouse is definitely different from the field, but a lot of the daylight changing and the diurnal change of the crops can be regenerated in the greenhouse as well, and we observed that.
So, okay, so in the greenhouse, do you turn the lights off at nighttime?
It depends on what we grow, but in the Purdue, most of the Purdue greenhouse, we follow the standard to, we tell the greenhouse manager, usually we turn on the light, yes, from about, let's say 7 a.m. to 7 p.m., and we turn it off over the night.
Oh, okay. Okay.
But for any research purpose, we can purposely turn on the light in the night and see some weird change of the plants, just for fun, maybe.
Yeah, that would be interesting.
Yeah. Thank you for the question. So I think I really need to move on. So here my point is imaging time really matters. Imaging time is very, very important. Unfortunately, now, around the world, so many people are flying the drone imagers above the cornfield. As far as I know, most of the flyers, they are not quite aware of this kind of the significant change. So that is why we need to do this work, and we need to publish our discovery to share the knowledge with people. And the fortunate part, the good side is we realized that the V shape is actually a very stable pattern. And this is over 1 month. Over 1 month, we can have all kinds of the weathers, right? Cloudy, sunny, cold, and warm days. We always observe a very stable V shape. That means it's actually not very difficult for us to calibrate this kind of the diurnal variance. I want to skip these ones because I'm overusing time, but the brief take-home message is, besides the imaging time, the other environmental conditions also matters. You probably remember we put a mini weather station by the field imaging facility as well, so we can do the correlation study. It turned up that the only thing is the humidity. Humidity is the only feature, environmental condition, which didn't impact our imaging result. All the other things like temperature, wind speed, and other things had a very significant impact in our remote sensing results. But we were able to calibrate that one. However, I have to skip this part. To move to the next one. Basically, what we can do with the facilities, after the imaging, we process these images. We can calculate something like this. This is something called a spectrum of the plants. Still remember, hyperspectral camera has hundreds of different colors for each pixel. If we connect all the hundreds of intensities together, you get this curve, which is named spectrum. And we can input this spectrum into some machine learning model, deep learning, PLS, support vector machine, you name it, all kinds of the machine learning multivariable training machines. And at the same time, you can sample the leaf and measure the ground truth, the reference data, such as nitrogen, water, and things. So you can see if you see some correlation between this spectrum and your measurements. If it shows some correlation, you can use a model to use your image to non-destructively measure nitrogen and water. So these are some of the modeling results. Many years ago, we did this modeling, just want to share with you. So for example, we can do the biomass. Basically, biomass is the fresh weight of the shoot. So between the x-axis, which is a ground truth, right after the imaging, we cut the shoot and weigh them. So that's how we get the x-axis. The y-axis is the prediction of the fresh weight of the hyperspectral camera. So you can see we don't need to destructively measure the weight. We can just take a quick scan. We know how many grams, grams is each shoot. And similarly, we can measure the nitrogen content in the plant, which is really important because the farmers want to know, do I need to fertilize or not? And the water content, leaf moisture in the leaf as well. And the next slide is just one breeding project example, how we use these prediction models. So for example, if you get a very good relative water content prediction model, you can do this kind of study. So you can grow the plants in the greenhouse until some stage, and then suddenly you stop watering. And what the plants will become? They will start to show some wilt. And some yellow leaf will showing up, right? So the hyperspectral, in this chart, the x-axis is the number of days after the drought stress treatment. The y-axis is the predicted relative water content from the hyperspectral imaging. So this is a non-destructive, continuous monitor of the leaf moisture change in this plant. So as you can see, on day 0, we suddenly stopped watering. So you can see the predicted water content is keeping dropping as expected. So the hyperspectral imaging, yay, it works very well, right? However, each color is a different genotype. So you can see the red, blue, and the green. These are the 3 different genotypes. You can see these 3 genotypes are losing water in a different speed. The green one is losing water with the fastest speed. You can obviously tell that, right? What does that mean? That is a bad genotype when our field is under drought stress. So in the very hot July or August, without several weeks, if several weeks without raining, our soil will become very dry. And it is not a good genotype for this green one. It's not a good one to be planted in that field because they will die first. But the red one and the blue one probably will survive and survive until the next rain comes. So just to show you how these facilities can be used by our plant scientists and breeders at Purdue, in and outside Purdue, to use these phenotyping facilities to do the data-driven, quick phenotyping, to look for the drought resistance of the genotypes, also looking for the higher nitrogen usage efficiency. And of course, we can predict diseases, right? So we can find those seeds which are more resistant to the pathogen and insect and chemical spraying diseases similarly. So very cool, right? So far. However, the however moment comes, okay? So since we already developed so many hyperspectral imaging and whatever other imaging sensors, as today, if you, let me quote something. So for example, the McKinsey, McKinsey is a big consulting company in Boston, right? Very famous. The McKinsey Global Institute issued one report and claiming agriculture is the least digitalized industry among all the major industries. And you were like, what? We just listened to Dr. Jian's seminar. He said, fancy technologies, fancy sensors and digital tools we can use for agriculture. But why McKinsey is saying this is the least digitalized? Because McKinsey is actually right. Although we have developed so many good, cool technologies, they are not being used right now by farmers. And I can quote another one. So the American Federal Bureau of Agriculture also estimated that by 2020, the market for drone remote sensing imagers is already surpassed $10 billion just for North America. However, yes, we did observe a lot of the drone imaging companies started up over the last 10 or 5 years. However, we are not seeing any of these companies growing up, doing more and more businesses with farmers. Instead, just 2 years ago, during the pandemic, we heard that in California, one of the biggest drone agriculture crop sensing company was shut down. It's because it has no business to do. Nobody is buying the technology. Why is that? So for the next 3 or 5 minutes, I'm going to share some of the technical insights of the current status of the application of the sensors. Basically, the sensors are all good, but applying the sensors in a very complicated agriculture scenario, the environment is the reason. So let's take a look at this picture on the top left corner. When we fly the drone above the field, again, we just talked about the lighting variance, right? The sun is keeping moving, the color intensity, everything is changing. The clouds are moving in the sky. So we're actually imaging the crops with a very unstable lighting source. So the ambient light is a big, big issue. So you can see the shadows of the clouds are applied in the field. So this is, of course, impacting our results. Again, imagine, can you still remember that spectrum curve I just showed you? We are predicting the tiny little changes of the color in the crop and using that information to predict nitrogen deficiency and diseases. But the cloud is applying such significant impact to our color information. And I can tell you, this is very difficult to calibrate. Many people put some white references, gray references, something like that in the field. But I can tell you those methods cannot calibrate this kind of the impact in a satisfactory level. So lighting condition is one big issue. And then let's take a closer look at the field. Let's take a look at this picture on the right side. The canopy of the corn or soybean field is a mess. It's not a uniform surface, it's a mess. Why it's a mess? If you take a closer at this canopy, you see a lot of the shining, very bright regions. These are actually the specular reflection. These leaves are just working like mirrors. You're actually looking at the sun through the leaves as a mirror when you're looking at these very bright spots. So you are doing sun phenotyping, not plant phenotyping even. On the other hand, you also see some of the very dark regions in the field. These are the shadows from the other neighboring leaves. So we have the very bright specular reflection and very dark shadows. And this difference is not of our interest. We are more interested in disease and nutrients, but this is simply caused by the environment. Also, you know, for the drone imagers, if you can have something like 5 inches of the spatial resolution, 5 inches is like this length, right? 5 inches of the spatial resolution. Nowadays, people claim that is the high resolution, spatial resolution for drone images. But with 5 inches, there is no way for you to clearly separate the soil background from your canopy. So you are doing soil phenotyping, not plant phenotyping. Again, finally, this is probably something not everybody knows, but as an imaging scientist, I want to share with you the slope, the angle of the leaf is a significant. If something is important, I say it for 3 times. Significant factor for imaging result. When the slope is changing, the image color, the color of the hyperspectral image is totally changed. So, and you know, every time in the field, the wind is keeping blowing, so the leaf is keeping changing the angle. So we cannot control this as well. And another thing is, again, with the drone imagers, you don't have the resolution to look at the details, but the details on the leaf is actually giving us a lot of the useful information. So for example, if you compare between the healthy leaf, the potassium deficiency, you know, the K deficiency and the N nitrogen deficiency leaves, when the leaf is losing nutrients, the symptom actually show up as different patterns. When it's losing K, the yellow leaf starts from the edge of the leaf. But when the leaf is losing nitrogen, the yellow starts from the mid-rib, around the mid-rib. This is very, very important information to tell us which nutrient is the problem, so you are not dump the wrong fertilizer into the field, right? However, with remote sensing, you don't have the resolution to tell this difference. Right, this is a typical scenario. Let's say with phenotyping sensors, one customer someday comes to the facility and asks, hey, could you predict chlorophyll content of this plant? And the very smart Star Wars style computer of this facility will ask back one question: which leaf are you talking about? Why the smart computer will ask that question? Because the leaf-to-leaf difference is huge, again. Leaf-to-leaf difference is huge, and even the regions on the same leaf is totally different from each other. That is why I'm not a big fan of these spot imagers, which is measuring a small spot on the leaf. I used to be at Pioneer, and in our Pioneer greenhouse over summer, I observed that Pioneer company hire a lot of the summer intern high school kids. So we use the school bus to carry dozens of kids to our greenhouse. And we spend a couple of hours to train them to use some sensor. This kind of sensor is called speedometer, which is a chlorophyll greenness measurement sensor. We train the kids to measure the field, measure the crop. And of course, we tell the kids which leaf to measure and where on the leaf to measure. However, if your measurement spot on this leaf just move 1 inch to the left or to the right, the measurement can be up to 50% variance between just that. How can the kids control the position so precisely like within 1 inch? So that is another problem. And this shows you more details. So we can use the hyperspectral camera to show you the detailed spectrum difference You know, it's on the same leaf. You just pick 3 different locations on the leaf. The spectrum is totally different. This difference is much bigger than the difference between a healthy leaf and the diseased leaf. So if your noise is stronger than signal, you cannot do the good phenotyping. And we talked about the diurnal change. The imaging time matters, but unfortunately, most of the current drone imagers are not paying attention to this one. So some people are flying today at 10 a.m. tomorrow because in the morning it rains. So we need to wait until 3 p.m. to fly. And you got that diurnal difference in your image. I will skip this one. This is a little complicated story and I don't think I have time. I actually have only 15 minutes left. I need to hurry. Okay, I will jump over because I really want to show some Purdue solutions to keep people happy when you leave this room, right? So first one, just want to highlight one of our new technologies called the leaf spec. So basically leaf spec is a handheld hyperspectral leaf scanner we developed at ABE at Purdue. So why do we need to develop this one? Because here is the thing. So, you know, we just talked about the facilities. We spend so much money to build these facilities, but these facilities can only benefit Purdue researchers. We have, at the same time, we have 600 million farmers around the world. They cannot benefit from our advanced technologies. So the question is, can we miniaturize the technology from the big imaging tower to a small handheld device? Basically, can we put the advanced technology into people's hands so people can measure anywhere in a growth chamber, in a greenhouse, in their own field, just working like this. So the good news is we did that. So in 2017 or 18, we developed this leaf spec sensor. It's a real hyperspectral scanner. It takes about 3 seconds for you to scan this corn leaf. And immediately it's processed and you can see the nitrogen, water, chlorophyll result on your smartphone app. And each measurement is georeferenced. Of course, why not? If the phone is already evolved in this loop, the phone has a GPS, so why don't we also remember where you measured this measurement? And again, this is a real hyperspectral imager, so you can see this beautiful hyperspectral leaf. Compare that with the existing sensors images. It is having the very, very fine resolution. You can see the midrib, you can see the veins. And each pixel here is having hundreds of different colors. This is a real hyperspectral image. So we can carefully analyze how the nutrients, the nitrogen, potassium, and the other nutrients are distributed on this leaf to give the more detailed information of the nutrients status and also the earlier detection of the nutrients. And by the way, I really want to mention this. Just now, I just shared with you a lot of the problems, the noises in the field, right? Have you realized that with this kind of imaging mode, most of the noises are gone? The ambient lighting is gone because we capture the leaf and scan. The leaf slope is gone because we keep the leaf flat and we scan. Then we have a much higher resolution, so we can do the distribution analysis. Everything's gone. So that was why 4 years ago or 5 years ago, we got so excited about this idea. And we luckily, we got a very good team, Dr. Wang at that time, and some of the very talented students joined together, and we were able to execute this plan and develop this device. And so far, 5 years later, we developed a series of different versions just because in phenotyping, there is no one-size-fits-all sensor products. We have very big and long corn leaves, but we'll have very small grass leaves as well. So we need to develop different versions to fit the different plants and for different purposes. And for some of the images, we can give the resolution like this. Looks beautiful, right? But of course, making the images looking beautiful is not our goal. Our goal is to get more texture information so it can tell us at the earliest time point when any disease starts to show up or when any nutrients start to show up. And again, because this is georeferenced, so this is how it works. You know, we can scan, some of us can scan in the field, and the evil professor like me can sit in the air-conditioned office And watching on my computer screen, I can see the dots are keeping showing up, so I know my students are working. But this is a joke, right? But the useful thing is we have this data infrastructure, the GIS map, the digital map to map our imaging results and basically tell us how because every time when you apply this kind of sensor, the IoT, basically the ag IoT sensors, you need to know the location question. Yeah, that's a great question. Brief answer is yes, definitely. Depending on which nutrient, which disease you are talking about, but in almost all the cases, the color changing patterns, the texture, and all the even the morphological shapes, they show up as different symptoms between the different leaves. So it is very important for you to know which leaf you are scanning, and then you need to have some protocol. So for corn scanning, for example, our general protocol is we scan the so-called top matured leaf or top colored leaf, because the color is a sign of the maturity for corn. We need to stick with that protocol, so that's really important. But of course, you can use this sensor to scan all the leaves and combine them together to do kind of the spatial series analysis as well, if you have time and throughput to support that. So, okay, this is just one slide to share the information that this product is successfully commercialized. So we started one company in 2018, and we have 8 patents applications. And the very good news is just last month, one of the most important patent application, we received the allowance letter. So basically we got that approved. That will really keep Purdue a unique place for this kind of the proximal sensing. And we are also working with a lot of the industry companies to apply this technology. So I have 5 minutes left. Just want to quickly show you, maybe you can pass this around. So we actually 3D printed the chlorophyll distribution. The chlorophyll distribution of the 2 soybean leaves, one is high nitrogen, one is low nitrogen. So usually with remote sensing style, you can only tell the difference between these 2 leaves when the yellowness, the general yellowness on the leaf is showing up. But with leaf spec, we can detect this kind of pattern at a much earlier time point. So nitrogen prediction, I'm going to skip this one. Same thing for corn. We can do the same spatial analysis of the nitrogen distribution. Thank you, Dr. Yu. Yeah, we can pass this around. So maybe I will pass this one. But this is actually a very important thing. This is basically the dicamba herbicide spraying thing. Dr. Yu, I think I'm overusing the time. I think it's better for me to show the robots, which is more interesting. And if we still have more time, we can come back to these applications. So basically, these slides I'm skipping now is just a bunch of applications, how the leaf spec can be used for detecting the diseases earlier. But the very interesting thing is this year, we also developed some robots which can use the robotic arm to automatically do field phenotyping in the field. So like this robot is named PhenoB. I hope this video is playing smoothly on Zoom. Let me know if you can see this video okay. So every time the drone can be programmed to fly automatically to the designated locations in the field. And every time it lands, and again, it uses machine vision to detect the top matured soybean leaf. And then the robotic arm will be, what it's holding is just one soybean version of leaf spec. So it captures the leaf, closes the chamber, scans the leaf. And immediately after the scanning is finished, you can see the results of the prediction of the disease or any herbicide damage or any nitrogen, phosphorus, potassium deficiencies on the smartphone app. And this one is, again, uploaded to our digital map system. And then the drone can fly to the next location. So this is a PhenoB. Another robot, this is just to show you how we're also doing some path planning, because with a drone, you know, we can control where we fly to and how we collect this distributed information. This is a video for corn version as well. So we can develop a robot carried by this kind of the Pheno robot. And similarly, every time it can detect the top matured corn leaf, and the robotic arm will hold the corn version of leaf spec to capture the leaf, move to the base of the leaf, close, and scan. Same idea, right? However, you know, with these robots, there is always a problem. Just like those, you know, if you go to the ag engineering conference, you see all kinds of the presentations on apple picking, orange picking robots. They use machine vision to detect the oranges and then use a robotic arm to pick it, right? So for most of the robots, you know, no offense to those robots, the apple picking robot developers, but I myself have this feeling every time I see the video, because these robots are kind of still slow, the throughput is still one issue. Every time I just cannot help to kick away the robot and do the picking by myself, right? I can quickly finish the job by myself. So similarly, we have the same issue with those robotic arms as well. Sometimes it's not a big issue. So let's say if a farmer give me a call and say, hey, Dr. Jian, I suspect I may have some dicamba herbicide drifting issue, which is a very important issue nowadays in the Midwest, okay? So he's suspecting that his neighbor's herbicide spraying is drifted to my field and my crops are susceptible to that herbicide. So I doubt if that happens or not. Can you use leaf, and our leaf spec, actually I skipped those slides. Our leaf spec can detect dicamba very accurately. So now the question is, now this farmer has 3,000 acres. This is a huge, huge farm, something like 10 of our Purdue campuses, okay? Of course, we cannot scan all the plants, but we need to use the drone to quickly fly to the different locations. And in that case, every time if you give me 1 minute, I scan one location with just 1 minute, that is okay. But in another scenario, let's say the Monsanto and Pioneer researchers, the breeders, they want to finish the phenotyping of their research spots of hundreds of different mini plots, and they really need to finish that within a couple of hours. You really need to be much, much faster. So that is why we developed this so-called proactive type of robot using a vacuum machine. Instead of using a robotic arm to catch the leaf, we use the vacuum machine to suck the leaf into the imaging. And we can do that with a much, much faster speed. So now just take a look at this one. So as you can see, when we still use machine vision to approach the approach the canopy of the top matured leaf. But the vacuum machine can keep this leaf very flat in a very good status to be imaged, and we alternate through the different colors. By the way, this is a multispectral imaging, so that's why you can see the different colors. So next slide, please. So this is the future, right? So we can use a drone to carry this kind of the vacuuming imaging machine. And of course, this is still the imagination picture, but for people who know me, you know, when we imagine something, we always deliver. So probably next summer, we will give some demonstrations of this kind of the fleet of the robots. Surveying the whole soybean field for dicamba 2,4-D drifting for Indiana farmers. Yeah, I think I want to leave some time for you to ask questions. So I will skip the next two, which is one is smartphone-based kit and an app, just to fill the low-cost market, but still a professional phenotyping market. And the next one is a root phenotyping imager. So basically, we basically developed something like a robot earthworm to carry a micro camera to crawl inside the soil to do mini invasive root imaging. And actually the result looks very good. We can recover with a mini invasive robot, we can recover the spatial the 3D structure of the root. And also we showed it's having very good signal of the drought stress, and the corn rootworm damage is also very significant with this robot. I don't think I control the time very well today. I missed something, but I think I will stop here, and thank you for attending. And I'm now ready to answer any questions. Thank you.
One for and then one for talk. Okay, you have any questions here? So maybe I can start with one question. Yeah, it's very exciting, very inspiring talk.
Thank
so
you.
Lastly, you mentioned the like worm logos to get to the ground and get some data. So you also take the just image data, or you can also,
Yeah. Basically, the idea is we use this robotic arm. This robotic arm is holding this probe, and at the tip of the probe, there is a very small micro camera and lights. So we insert this probe into the soil, and then you can imagine it's kind of like the rootworm is crawling in the soil. And we keep taking images at different locations, 3D locations in the soil. And every time if there is a root beside that tip, this machine vision algorithm is a key. So for over 96% of the chances, we can detect that root if there is a root. And imagine we keep inserting, and we detect that here is a root, here is a root, here is another root. Eventually, we can accumulate this kind of the cloud, point cloud of the roots. And then we can use some kind of the neighborhood connection algorithm to connect them into the original 3D structure of the root. And then you can measure the width, number of branches, the length of the branches. So especially for the drought stress tolerance, one way for the corn plants to be drought stress resistant is to grow the root deeper and deeper, because that makes it easier to still find water in the deeper level of the soil. So basically, just for your information, nowadays in a breeding company like Pioneer and Monsanto, for drought stress is so important for breeding, right? Basically, two directions. One is on root, like what I just mentioned. Another one is to find the plants which can automatically close the stomata on the leaf, so to stop the transpiration, to save the water when it is under some drought stress automatically. So Jian is like magic, right? Sometimes. Did I answer your question?
Yes. Maybe I can ask a follow-up question.
Sure.
So here is like using this robot probe to detect whether there is a root or not. I just wondering, maybe also you can use the image data to tell if based on the color of the root, if the healthy or is what condition of this plant.
Yeah. Thank you. So that's actually a great point, because we not only know the location of the root, but also we got the images of the root. And one typical example is, for example, in this slide, we did one project with the Western corn rootworm. Some of you heard about this. This is a big issue for Midwest of the United States, right? So if there is Western corn rootworm in the soil, they usually, if this is a seed area, the root grows from the seed, right? So the corn rootworm is usually in the area right underneath the seed, in the thick stem of the root. So, and if the root is infected by the corn rootworm, you dig them up, and if you look at that section, they look yellow and brown and black sometimes. So yes, color information sometimes is important. So actually, our students successfully combined the color information and the morphological information together, and reported a very sensitive signal to the disease. Thank you.
You have more questions here? Let's see. Do you have questions online?
Do you have questions?
Yeah. Yeah.
So maybe I can try to ask a question.
Sure.
Yeah. So thanks, Dr. Jian, for the insightful talk. So in control, we sort of talk about closing the loop. Right? So I think your talk sort of focus on the sensing aspect. There's also actuation.
I love the question.
The benefit of
Yep.
For the sensing purpose, of course, you want to get as much information as possible. in reality, probably, the more information you're getting, cost higher. And it's also possible that in production, it may not be easy to utilize some of the information. Right? So this value of information should be saturate at some point for the purpose of driving production value. Do you have like any sense about how much and what is that sweet spot?
Okay.
Yeah.
Okay. I think that's a great question. So you are basically asking about the challenges in maybe data storage, data volume, processing power.
More about different scale technology because really cheap cost and easy to scale it up.
Yeah.
And meanwhile, all the information there should be translated into some decision for the box.
Yes, yes. So that's why it's called closed loop, right? I love that question because this is actually my career dream. I actually gave some seminars just in the last several months. And if people ask me, hey, what is your career goal within, let's say, 10 years, 20 years, I usually say my goal is to close the loop. Because talking about robots, this is just the sensor. We still need to have the computing unit and we need to have the actuators. So this is also why I need to go back to the two quotes I just mentioned with the McKinsey Global Institute, with the and also with the American Farm Bureau's estimate of the market. There is a huge market for this one, but it is not filled in yet, just because we haven't closed the loop yet. We only have the sensor, and the sensor doesn't help people. The beautiful image doesn't help the farmers. The farmers need to know, tell me the answer. Do I really need to put the nitrogen here or not? So this is exactly what we're going to do. So for example, now at ABE and some of the other agriculture departments, including some of the other engineering departments, for example, we are right now writing some new proposals, trying to lock down maybe $10 million or something to build an experimental site for this kind of closed loop experiment. Without details, but roughly the idea is, these are all I talked about today is sensors, but in a typical precision agriculture, the actuators are probably the sprayers to treat the diseases, the fertilization, and also the irrigation. When and where and how much should I put water, nutrients, and chemicals into the field? These are the decisions to be made. And right now, what farmers are doing is partly they are looking at the plants with their eyes. Partly they are listening to the weather stations forecast, because sometimes when the moisture is coming, they know some diseases are going to show up, so they spray. And sometimes they listen to the experts' suggestions. And sometimes they take the leaf samples and send the leaf samples to some of the wet labs to analyze the leaf for those. But that is too slow because it usually takes several weeks. In other words, we have very good sensors, but these sensors are not included in the closed loop applications. But I really see the light at the end of the tunnel. I think part of the reason that after developing sensors for so many years, we are still not in a closed loop is because of the quality of the sensor signal. This is my personal opinion. This relates to some of the noises I just shared with this group today. Because of the so many noises are existing, we are suffering from this signal over noise ratio issue. So we need to calibrate all those noises. But at the same time, we also find some kind of the shortcuts. So for example, leaf spec is like a shortcut which quickly removed all those noise issues. So I think it's worth a try now to really build some facility. So with this facility, we have not only sensors, but also actuators, the spraying, irrigation, fertilizing machines, maybe on a central pivot in a farm. And also we should have computer have a server standing in the middle to close this loop, so that let's really have a comparison. Let's say we have a control farm. We do whatever farmers are doing in the control farm. And we use our new sensors to decide on where and when to fertilize and to water. And finally, let's compare who has the bigger number of bushels per acre in the farm. And can we wrap up the sensor and the computing units into commercialized products to reduce the cost? Hopefully, after mass manufacturing, we can reduce the cost. So finally, with the cost and with the yield improvement, we can prove that this mode can help farmers to make a profit. That is a golden standard. Money is a golden standard. So that day, I would say, if before my retirement, I probably can still work for 25 years, but before my retirement, if that can happen, because if the American Farm Bureau's estimate is true, in digital agriculture and in precision agriculture, we should have a company with the size of Google, Apple, or Amazon. We should have a company like that. It does not exist now, but if our sensors can make that happen, and of course, most importantly, can help the farmers to save the time and make the right decisions. That is why we're working on this. But I love this question. Yeah.
Great. I think that's all questions for now. Let's thank our speaker again. And, thank you very much. Yeah,